Okay, and we're back. So, welcome back to this time we're doing lecture 3. I will actually be splitting this apart into two lectures because one part focuses on reactions which occur in glycolysis and gluconeogenesis, and the other one focuses on ATP and other high energy molecules. They do have some similarities, but I'm going to split it apart. So, there'll be a part 1 that's up already, this one will be part 2, and then the next one will be part 3 of bioenergetics. So, let's get started. So, bioenergetics, like I said before, is the amount of energy used in or released in a reaction, right? Let's go over certain reactions which are common in glycolysis, gluconeogenesis, and other reactions in the cell. So, there's six main reactions which are used throughout. This includes a redox reaction, which is an oxidation reduction reaction, a group transfer. This is glycosyl, acyl, and phosphoryl. You have hydrolysis reaction. You have a carbon bond and carbon bond formation and cleavage. And then you have free radicals reaction. And the last one is internal rearrangement and isomerization and elimination. So that's a quick rundown of all the reactions that are used either in glycolysis or gluconeogenesis or both, right? There's a mixture of them. We'll cover that in the next lecture. So right now we'll focus on what each reaction is, what is used in each reaction, what's the general product made. Okay, so let's get started with probably one of the easier ones is internal rearrangement, isomerization, and elimination. So in terms of internal rearrangement and isomerization, this focuses on, let's say, a compound of glucose or any other carbon compound. It's an oxidation and reduction reaction which occurs within the molecule without any external stimulus or external help. So pretty much one part of the entire molecule gets oxidized and another part gets reduced. This forms a whole new molecule without any external help at all. In terms of the elimination part, it just means the elimination of a water molecule to form double bond. That's reaction number one done. The next reaction we'll cover, which was talked upon in the last one, is a redox reaction, which is also known as an oxidation reduction reaction. It was covered in metabolism, like anabolism and catabolism. Right? It was in the last video. If you haven't watched it, be sure to check it out. Great video. So this one is more of like the more common, more well-known reactions. A redox reaction has one object that's being reduced and one object is being oxidized, and in the product they flip. One will be reduced and one will be oxidized. That's pretty much it in a redox reaction. The only difference is it uses n electron carriers. Sorry. So an electron carrier would be NADH and FADH2. And like I said before, I don't remember if I said it, but I'll go over it again. So NADH and FADH2 have two forms, an oxidized form and a reduced form. NADH is a reduced form, FADH2 is the fully reduced form. The main difference between these two are they require they use the same amount of electrons, but FADH2 holds on to two of them, NADH holds on to one of them, and the electron that's left it will go to another carrier, right? So that's pretty much the main difference between those two. There are other electron carriers, so NAD plus, NADP plus, FMN, and FAD. These are all the four main water soluble coenzymes or water soluble electron carriers, which are these ones specifically can undergo redox reaction because they can go reversibly between oxidation and reduction, more commonly known. But there are other ones which are used in different reactions. So like you have your keen ones, which are lipid soluble and carry electrons in the membrane, like the electron transport chain, right? So you have your ubiquinon, which is using electron transport chain, and your plastocane one, which is used in the plants, which we will not cover. And then the last one are your prosthetic groups, which also undergo reversible oxidation and reduction. These usually are used in the electron transport chain as well. So iron sulfur proteins and cytochromes, which is you guys should know from Bio 1 or Cell Bio, right? Those are pretty much the two reactions. They move electrons through the electron transport chain, right? Through these proteins. So those are the main electron carriers. And then another thing you need for a redox reaction is a reducing agent and oxidizing agent. Like I said before, one molecule will be reduced, one molecule will be oxidized. That way it's easy and that way it's balanced and not a lot of energy changes. Another thing you need is a dehydrogenase, which is just an enzyme. Usually it could be complex or not, but a dehydrogenase is used in redox reactions. So as soon as you see that it's a dehydrogenase complex, you know it's a redox reaction. Great. So that's pretty much it for the redox reaction. There's not much else. We'll move on to the next one, which is generally a little bit easier, is a hydrolysis reaction. Many of you should be familiar with a hydrolysis reaction, but we'll go over it, right? So a hydrolysis reaction is utilizing the addition of water in a compound to break apart a covalent bond and get a new product, right? So in terms of like, let's say example ATP, if you intern if you add water to an ATP at the gamma site, the gamma phosphate will get, um, sorry, the gamma phosphate will get cleaved off and you'll be left with ADP plus water, right? And so ADP plus water then can undergo ionization and then turn into more stable form ADP3 minus. And then the last phosphate group, the gamma phosphate, will undergo resonance stabilization to turn into a phosphate group with an inorganic phosphate group which can be used in other reactions, right? So this is like a highly exergonic reaction, which is used fairly often if you need to fulfill an energy requirement for endergonic reactions. So the next one would be another easy one is carbon bond cleavage, right? Which is just the breakdown of the covalent bond between carbons, right? So there's a carbon, you get the point. There's carbon, two carbons, right? And it's carbon bond breakage. And so this is pretty much just two different types of cleavage, heterolytic and homolytic. Heterolytic is the unequal distribution of electrons that make up the bond, covalent bond between two carbons. So that means one of them will get two electrons, the other won't. This results in a carbon cation and a carbon anion, which is important because they're used in different things, but they are fairly unstable, so they're not always like that. And then the other one is a homolytic. Homolytic is the equal distribution, so right down the middle. This results in free radicals, which isn't a good thing, but it's not a bad thing because it can be used in a different reaction we'll cover. But that's pretty much just a little bit later, and then we'll cover that reaction a little bit later. But that's pretty much it for cleavage. You can also have a decarboxylation, which is the release of CO2. In terms of carbon-carbon bond creation, pretty much aldyl condensation and Claisen ester condensation. That's pretty much it. You can also have a nucleophilic attack via a carbon cation, but that's pretty much it. A carbon cation attacks the electrophile. You get a carbon-carbon bond. So we went over internal rearrangement and elimination. We went over carbon bond and carbon break, redox, um, hydrolysis. We'll go over free radicals and then group transfer. Group transfer might be the more complex one because we haven't done anything like that so far in biology. We'll do free radicals. Free radicals, you take your homolytic cleavage, right? The equal separation of electrons. You have two. You're gonna take it, and typically it wants to pass along the lone electron to a larger compound, right? So if you have a carbon chain, you want and you have hydrogen available, the free radical will take the hydrogen, pass the radical onto the carbon chain, and then it will undergo double bond formation to stabilize it and get rid of the radical. That's pretty much it. The last one that we have, right? The very, very last one that we have, and honestly, one of the more confusing ones is the group transfer reaction. So, like I said before, there's three. I don't. How do you hold up three? Three, 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 three. Anyways, there's three different reactions associated with a group transfer. There is a there is a glycosyl, phosphoryl, and acyl. By far, the acyl one's the easiest. Then you have your phosphoryl glycosyls. A little bit hard to get your head around, but it is what it is. So start with acyl. An acyl group transfer really easy. You have an electrophile. You have a carbon acetyl group, right? You get. So here's your acetyl group. You have your electrophile. Electrophilic. Sorry, you have your neutrophile. Your nucleophile. So you have your acyl group, you have your nucleophile. You're just gonna take the nucleophile, do a nucleophilic attack at the central carbon. This causes the breakage of the double bond, tetrahedral intermediate, and then after leaving group leaves, you're left with your nucleophile addition plus a double bond again. That's it. That's it. That's your acyl group transfer. Your nucleophile enters the acyl group, tetrahedral, tetrahedral intermediate, and then leaving group leaves. That's your acyl group transfer. We'll move on to a phosphoryl group transfer. So, as you know, we'll cover this more deeply into the next video, but we have ATP. As an example, ATP has three different phosphate groups. That's why it's adenosine triphosphate, three different phosphate groups. They all have different products and different names. So you have alpha, beta, gamma. We will cover this in the next video. But for this example, you have a phosphoryl group transfer, which pretty much just means that let's say you have a glucose molecule and you have ATP. Your glucose molecule will steal or attack the phosphate group, let's say gamma phosphate, and it will break the phosphate group away from the other phosphate groups in ADP or ATP, turning it to ADP. So what you're left with is ADP and a phosphoryl group attached to glucose. That's it. Straight and simple. The last one, and so that's pretty much it. In that specific reaction, you start off with ATP, you have glucose, you're left with ADP and glucose 6-phosphate, which is used in glycolysis. Step 2 of glycolysis. The last one, maybe the more confusing one of the out of all of them, is a glucosyl glycosyl group transfer, right? So a glycosyl group transfer, similar to a acyl group transfer, the only difference is you start off with glucose, you have a nucleophilic addition causing a breakage of bonds, and you have a glycosyl carbon cation intermediate. Then you have another nucleophile attack from the bottom, create a new bond, and you make glucose. That's pretty much it. Yeah. So that's it for all the reactions. Like I said before, we're gonna split this apart into two videos. So there'll be three total videos of bioenergetics on the channel. The first one covered the different variables associated with it and the additive principle. The second one, which is this one right now, covers the different reactions used in glycolysis, gluconeogenesis, and the different things used in each. And the last one will cover ATP, which is an important molecule in pretty much any reaction in the body. So, thank you guys for watching. We'll see you in the next video.